All right, last environmental factor we're going to discuss here from chapter 6. Do you have neighbors? All right, so living things across the planet, not just microbes, can live with different associations. Some of this terminology you've probably heard before somewhere back in school. First of all, there are relationships that are called symbiotic, and this is where two organisms live in a very close relationship with each other. That relationship is nutritional, and it's required by one or both members. Okay, so there are three categories. One's called mutualism. Mutualism is basically where both members are benefiting. Commensalism are both members are benefiting, and it's generally required or obligatory. That's a mutualistic relationship. Commensalism is where it's required for one organism or the organism is benefiting. The other one is not necessarily benefiting, but it's not being harmed from the relationship. And then finally, the third one, parasitism, we've talked about this before. This is where the parasite is dependent upon the host, but the host gets harmed. Okay, so hopefully you can see the differences between these three different categories. How about our normal microbes? The normal microbiota, the normal microbes that live on or inside the human body. Well, hopefully those are not parasites when they're living where they're supposed to live. But are they mutualists or are they commensals? Well, for a long time, most microbiologists thought those organisms, like the E. coli down in your colon, were commensals. They were in this category. They were benefiting because their nutritional needs were being met by living on and inside our bodies, but they weren't really doing much for us. But the more and more that we study these normal microbes, there's a big explosion in research into those organisms. We're learning that many more of them are probably mutualists, which means they're benefiting, but so are we. They're doing nice things for us. They're protecting us from pathogens invading the parts of the body where they live, or gut bacteria, bacteria down in the intestinal tract do nice things for us, like manufacture vitamins that we can't make for ourselves. You know, it's even more and more becoming clear that you need the microbes that live on or inside your body for your immune system to function properly. That's a hot area of research. So it's probably going to turn out that a lot more of these microbes that live on or inside our body are mutualist, and we're dependent upon them just like they're dependent upon us. We got to have them. All right, and then there are non-symbiotic relationships. Uh, these are relationships where organisms may live together, but it's not required. And if you're living together and you're helping each other, that's called synergism. If you're living close to each other, but one or both are trying to inhibit or destroy the other, that's called antagonism. And again, these are not required relationships, but they are relationships that can happen. So synergism, you know, if two microbes, for example, are living, or two or more are living in a synergistic relationship, they're cooperating, they're helping to protect each other, they're sharing nutrients. But it's not the same as mutualism over here, where they have to live together. So that's the key difference between something like synergism and mutualism. Notice this terminology over here too, when thinking about parasites. An ectoparasite is a parasite that's living outside the body, like if you had lice, something like that. Endoparasite, inside the body. Intracellular parasite is living inside a cell. And we've talked before about obligate versus non-obligate parasites. That means obligatory. They must do it, or they don't have to do it in the case of non-obligate. All right, biofilms, we've already touched on this already, but biofilms are an example of synergy. Okay, so this is not an example of mutualism or commensalism. This is a synergistic type relationship where microbes get together and they cooperate with each other nutritionally and other ways as well, but they don't have to do it. So they actually change their behavior when they sense that they're part of a community. And this is a hot area of research as well because it's been discovered that many microbes are capable of changing their behavior and forming biofilms. And these biofilms have really important implications from a disease standpoint and from a control standpoint, because if you start having biofilm development on things like catheters, that becomes a huge concern in patient health care and making it more likely that patients are going to develop infections. Okay, this diagram over here is showing you like a model of a biofilm. You got some surface here, like this could be the surface of your teeth, which is kind of unpleasant to think about. And notice you got clumps of bacteria that are adhering to the surface. And so what happens is these microbes start secreting these sticky substances. Many of them are substances like you would find in a capsule, the glycocalyx, but they're producing them in thicker, more abundant amounts. And these sticky substances help protect the microbes, as you can see there, and helps them adhere, stay attached to the surface. And it's been discovered that some of these biofilms are pretty elaborate in terms of the architecture that they have. There will be spaces that are available within the biofilm to allow water to flow and pass through. So that's going to allow things like oxygen gas to channel through there, and nutrients to channel through there, and wastes to be taken away. There's a process by which bacteria sense that they are part of a biofilm. Many bacteria can do this. It's called quorum sensing. Quorum, like if you have a quorum at a meeting, you have enough members at a meeting to actually cast a vote that binds. And it's the same thing with bacteria. Quorum here is referring to bacteria. Like if you have a bacterial cell here, you have some bacterial cells scattered around. Bacteria send signaling molecules outward towards neighbors, and if you receive those signals, the other bacteria will have receptors on their surfaces, and if they receive those signals, those signaling molecules bind to the receptors. If enough of that happens, the bacterial cells realize they're part of a quorum, or they're part of a community, and they'll change their behavior. Okay, so they'll start producing these sticky, carbohydrate-rich substances that are going to allow them to attach to a surface like you see over there. That's called quorum sensing. Okay, and your textbook has a pretty nice diagram, which is showing you the steps that it takes for bacteria to convert from a free-living form into a biofilm-type form. And they have kind of a disturbing diagram in the book where they're showing you Staphylococcus bacteria in blue. Those are staph bacteria. And in green, that's gauze. Those are gauze fibers like you would have on medical gauze. And the orange stuff is the sticky substances that those bacteria are producing to help them stay attached to the gauze, which is kind of disturbing to think about. So step one, if bacteria are going to form a biofilm, they have to arrive on the surface and start growing there. They're colonizing that surface. And step two, so once quorum sensing has occurred, that's what they're depicting here in the diagram. Oops, I keep advancing the slide too quickly. The little red dots there, those are signaling molecules that the bacteria are sending to each other. And when the bacteria receive enough of those signals, that triggers them to change their behavior. And that's what they're showing you in part 3. I just did it again. Once the bacteria realize they're parts of a community, they will start secreting in gray there sticky substances that help them adhere to that surface. These substances are surrounding the bacterial cells and protecting them from things like drugs that a patient might be getting treated with. Not good. From a getting over an infection standpoint. And sometimes, like they're showing you here on the diagram, other species might get involved in the biofilm. A biofilm is not necessarily just one species of bacteria. And a good example of that would be the bacteria in the plaque on your teeth. That's a complex biofilm that contains many, many different species that are there. Incidentally, the orange stuff on the diagram can be referred to as an extracellular matrix. So you guys learn about that when you're studying tissues in Biology 201. So you have cells, and then the surroundings around them, the spaces in there, in your body filled up with things like protein fibers, like collagen fibers that occupy that space. That filler space between the cells is called extracellular. Extracellular means outside the cell. Matrix. Same thing in a biofilm. All these sticky substances that the cells surround themselves with can be referred to as an extracellular matrix as well. All right, and then notice in part 4 of our diagram over here, the biofilm can be a constant source for new bacteria to break away and float off and colonize some other surface. So once you have a biofilm established, those can be a source for those bacteria spreading to other locations, such as other locations inside the body, if you have a biofilm that's involved in part of an infection. And I think I've already mentioned in class, kids who have recurrent ear infections, it's been found that they can have biofilms that have developed in their eustachian tubes or in the inner ear. And once the bacteria, like you see over here, have covered themselves up with these sticky substances, and you've got some drug like penicillin over here, it can't diffuse through that biofilm, those sticky extracellular matrix substances, very easily to get to those cells and kill the cells. So the biofilm actually helps make the bacteria more resistant to the drug. This is a cross-section through a catheter, a urine, like a Foley catheter that would be inserted into the urethra and bladder of a patient to help them urinate. You're looking at it in cross-section, like you're looking on the end of the catheter. And all that crusty purple stuff you see there, this is obviously very heavily magnified. All that purple crusty stuff you see there is a biofilm of bacteria that are growing inside the catheter. So imagine that's inserted into a patient, and bacteria are breaking off from that biofilm, and that allows them to spread to the inside lining of the bladder and maybe even deeper into the body from there. So biofilms can be a big-time problem medically. There's a YouTube video clip on this page if you want to take a look at it, which discusses some biofilm research that's going on at a university in North Carolina. So some researchers are trying to develop new types of drugs that will help break apart biofilms. You know, like I just said, if bacteria are in a biofilm, it's difficult to treat an infection when the bacteria have surrounded themselves with all those sticky substances. If we can come up with new types of drugs that tell the bacteria, break apart your biofilm, don't create these sticky substances, then maybe our conventional drugs like penicillin, amoxicillin, those types of things will do a better job at locating those bacteria and killing them. The gross teeth you see over here, that's just to illustrate that the plaque on your teeth is a type of biofilm. And if you had an electron microscope and you scraped some of the plaque off of your teeth and looked at it with an electron microscope, you would see all kinds of stuff in there. We might take a look at the plaque. We might scrape your teeth in lab and take a look at that under the microscope just so you can see some of the diversity of microbes that exist within that biofilm. All right, I'm going to stop this video clip here. I wanted to spend a little bit more time on biofilms. The next video lecture clip, we're going to finish off the chapter 6 content and talk a little bit more about bacterial growth.